It's like following a rock star right now, at least in the drug discovery world. So basically, Paul talked about the discovery of Tofacitinib from his perspective. I'm going to talk about some of the work we did on the backup program, particularly around the structure of the different JAKs and how that leads to possible ways to come up with new inhibitors for a number of different indications. So taking a step backwards, here is the kinase domain structure of JAK1. And this is just of the domain, it's not of the rest of the molecule, which would kind of go around here and down. Basically, you have the N-terminus, followed by a hinge region, and then to the C-terminus. And at that hinge region, it's kind of special. There's a very hydrophobic pocket that ATP, or in this case ADP, because that was done in this particular structure, binds. And when it does bind, I hate that glitch, when it does bind, that terminal phosphate is transferred to either a protein or in many cases another kinase. For instance, with JAKs, that's a STAT, and then that initiates the downstream event. So when we look at the kinase domain in a closer up view, basically you can see that it's a very hydrophobic pocket. It has pi stacking interactions with the ceiling and the floor, so it's a very narrow cleft. Typically, particularly when Paul's group was doing this, the inhibitors were very flat. They resembled ATP. They had cores like ATP. And usually the heterocyclic core would make at least one hydrogen bond to this hinge region, if not two or three, and that would help it maintain some affinity. And then basically then you would progress out. Because there aren't many great selectivity elements within kinases, particularly early, it was very hard to get good selectivity, and you often tended to build larger molecules. And so it was very hard in the 90s to come up with a compound that was truly selective. Mark Flanagan ran the chemistry group when Paul was a biology team leader. I'm going to talk just very briefly and just show a few structures to this crowd of how they pursued this project. They did a broad screening strategy and came up with a hit that was about 200 nanomolar, and that's actually incredibly a potent hit at JAK3 at the time. And here's the core that would bind to the hinge, and then it had a lipophilic fragment up here that really is a little too big to do much with. And so they did a medchem thing and truncated off a portion to get to aminocyclohexane. They have pretty good potency there. And then they turned to a natural product by looking at an analog of carvone. And why they did that, I'm still not clear to, it's not clear to me, but they basically made a couple of analogs replacing this with an amino analog of carvone, which has 3 chiral centers, 2 of which were resolved very quickly. And what they found is all of the activity presided in the 1 enantiomer, compound 11, both in enzyme and cell, and the opposite enantiomer really didn't have anything going for it. And that was pretty remarkable because remember when I showed you with kinases, it's a relatively flat pocket. So that here they have good stereochemistry right away, and that was something that was important, and fortuitously, they followed up with it. So they resolved to get this pure enantiomer here and improve potency a little bit more, and then they still had the problem of it's not a very tractable compound, so they were able to introduce a nitrogen here, and then replace this isopropene with various functional groups, particularly amides and ureas. Eventually that led to Tofacitinib and the discovery of that. As Paul alluded to, when we started to play around with Tofacitinib and looking for backups, we had revised models. Sorry, keep missing the pointer. So basically, we ended up determining that Tofacitinib was not truly that selective for JAK1, 2, or 3, and Paul wasn't too thrilled at the time, but fortunately the compound was in phase 2. The data indicated it was safe and working, so it wasn't really too big of a concern. More importantly, particularly for anemia, we have good cell activity and good selectivity against JAK2, which you would worry the most about for the GCM SF STAT5 pathway and the JAK2 homodimer, where you compare that potency to the JAK1/3 or JAK1/2 heterodimers, where you're about 10 to 20-fold more potent. So that's a good window over the anemia side effect option or possibility. Another thing which was striking that came out in 2008 is Ambit did a broad screen of a number of kinase inhibitors on the market and in development, and they did screen Tofacitinib at that time, and they screened against 200 kinases out of the whole kinome. Tofa hits the JAK branch pretty hard, the JAK 1 through 3 plus TYK2, which is the fourth member. It only hit one other kinase. So at that time, this was truly remarkable and unprecedented to have a compound that selective and that small, because the molecular weight of tofacitinib is very small. People in the past had to go to molecular weight 450 to 500 to get selectivity of that kind of ability. So it's a very unique compound in that perspective. So we were fortuitous to start to be able to get structures of tofacitinib against several of the JAKs. And shown here is it binding in the kinase pocket. Now, this is not tofa, this is a tool compound, and it binds in, makes 2 hydrogen bonds to the hinge, and it's a nice flat arrangement. In the floor, it's kind of flat, and the ceiling's relatively flat, and the compound fits in. And if this was your only structure, and you didn't have tofa, you might start to make analogs of it, and you would assume there's really nothing to do with the ceiling and the floor, and you just go further out like people were doing with kinase inhibitors. Fortuitously, we were able to get a structure of tofacitinib, and it does several things right away of significant interest. First, when we compare the structure of tofacitinib and I'm going to keep doing that apparently. Basically, when we look at tofacitinib in JAK1, JAK2, and JAK3, it binds the same exact way in all of the JAKs, so that's the same orientation. Another point, and this is even much more important, the compound it binds to the hinge, and it has that chiral group I've been kind of talking about, and that projects this ketonyl up to the ceiling, up to the G loop, and in doing that, it fills a hole which has an unfavorable structural water. And in kinases or proteins, if you have a water that's not really great for the energetics of the enzyme or kinase or whatever, and you can displace it with something that it likes better, you get a huge increase in enthalpy and a huge boost in potency. So that's incredibly important to be able to do that. Now I'm colorblind. And then also importantly, when tofa binds, it creates a hole in the bottom of the kinase pocket or the floor. And doing the combination, so you have a flat binding pocket, but you also have done that very close to it, and you kind of lock it in like this typical key in a lock type concept. And so that 3D shape really gives tofa a lot of its properties as far as potency and also selectivity versus the kinome. And finally, and I'll get to this in a minute, JAK3 has a cysteine at this location in the kinase. It's kind of a unique position, and all the other JAKs don't have that cysteine. And that's a selectivity element that I'm going to talk a little bit about as far as JAK3 selectivity when we get to that slide. So I'm now overlaying the first slide, compound 6 with tofacitinib, and I'm going to reveal that I did have one partial lie, is on the cyanomethyl pocket in the ceiling, that actually exists in this structure, even though there's nothing to fill it. So when compound 6 binds, it doesn't fill that pocket. That structural water is still there. That's not very happy. But, it's just a fact. But with tofa, it fills that pocket very nicely. It also, tofa, as I said, goes to the floor. And what it does is actually induces a hole in the floor by stabilizing the flip of a leucine that's there. And it stabilizes it in a conformation that allows the methyl of tofa to penetrate and lock it in. Again, so those two interactions are the key. And so the key lesson, I think, from this slide is that you want to think about the receptor flexibility when you do a drug design process. Kinases are very mobile enzymes. They have multiple conformation states, and you really need to consider that. But sometimes these minor movements can be incredibly critical and lead to the best bang for the buck. So here is a slide showing tofacitinib docked in the 3 different JAKs. It's docked in JAK1 in orange, JAK2 in blue, and finally JAK3 in cyan. And in all cases, the conformation is very much the same. You see in the ribbon diagram, you really don't see any significant large changes between the JAKs. What we have shown here are the different residues where there's at least one residue that's different than either of the other two. And so you can see those sites. The thing is, most of these residues, like up here, are pointed way above in the wrong way of the binding pocket. This is kind of pointed out, even though it doesn't look like it. Most of these are also away. The only one that's potentially close to the binding pocket for a small molecule is the cysteine and the serine that I had previously mentioned. So basically, this type of structure tells you that it's going to be very hard to get a JAK inhibitor that's exquisitely selective for only one of the JAKs. You do see flavors. Insight has a compound that favors JAK1, JAK2 over JAK3. Others have maybe the opposite, but you usually hit 2 or 3 of them and may spare 1. And that's been the typical industry for quite a while. Now, recently, people have started to think about JAK3 selective. As soon as we found out that Tofa wasn't selective, that was something we all concentrated on. And one thing, knowing that it's hard to select in any other way, we were really curious by the cysteine. And if you could come down to here and hit that cysteine, that would be really remarkable. And I've shown 2 structures here. They're actually very similar to Tofa, and then a vinyl analog of Tofa. And this basically contains an acrylamide. And acrylamides are known to be able to react with a cysteine and a protein under the right environment and form a covalent bond. In fact, there are drugs such as Tofacitinib and I'm blanking, ibrutinib. Thank you. And I should know ibrutinib pretty well. Anyways, so you can do that. In fact, those kinases have a cysteine in the same location. So what people have been doing have been trying to take advantage of it, and it's been a little tough. When this compound was disclosed in just last year, they do publish a structure of that I'll show in a second. But when you look at this and you're trying to do a computational model of this compound bound to the Tofa structure, and you do the minimizations that you can do, the compound actually doesn't bind the way you might expect. It binds exactly like Tofa, puts the vinyl almost towards the ceiling, puts its own methyl flat, and it doesn't do the interaction with the hole in the pocket because it can't, because the methyl's in a different place. But it doesn't seem like this would be a good irreversible compound. And 10 to 12 years ago, you might have been stuck with that. We have newer software now that allows one to handle this differently. So shown here is the actual structure from the Pfizer work done in Cambridge in 2016 on 665-1600. And this compound is another high-resolution crystal structure. The compound's gone into phase 1 for Crohn's disease. And what they see in the crystal structure is the compound has effectively flipped the piperidine and now moves the vinyl over to here, and that vinyl sits near that cysteine, and if it sits there long enough, the cysteine will react. And that's what you want. You want to have good affinity, and then you want to have the cysteine capture it. And so when they do that, they have a compound that's very selective for JAK3. It doesn't hit JAK1 or JAK2, and they've proven that in enzyme, which is easy. And they've proven that with some cell assays that are a little bit trickier, but they've proven that this compound truly is a JAK3 selective compound. And recently, Merck has also disclosed another JAK3 selective compound in a paper, and they both support the idea of what a JAK3 inhibitor can and can't do. And it's a very interesting paper to consider. So basically, what we did here, just to verify things, we did dock both Tofacitinib and the 1600 compound. And we did the 1600 compound in what we would call a covalent docking mechanism. Here you basically can tell the program that you want to form a bond with that cysteine. And these programs existed 5 years ago. They're a lot better now. And what it does is it varies the receptor and it varies the shape of the ligand. And it does this kind of in a multiple fashion. And it can usually give you a realistic result. And in doing so, when we dock 1600 in either the Tofa structure or the 5-TOZ structure, we get the same overlap. Basically, it binds like the X-ray structure shows. So we have software that can predict this pretty well, at least in most cases. So doing this and thinking about all of these structures, we have a lot more in hand to try and design new flavors of JAK inhibitors for different inflammation indications and dermal applications. And so the key lesson here is the ligand flexibility is just as important as the receptor flexibility. And then finally, I just want to conclude. The discovery of Tofacitinib took almost 20 years. It was a group of many, many people. Paul was the leader who helped start it at Pfizer. And you could say it was all perfectly done, but it wasn't, as Paul described. There was a bit of serendipity. And that's the way all drug discovery programs are. From that, the knowledge of cytokine circuits and dermal diseases, plus our growing knowledge of the structural aspects of binding to JAK inhibitors, should allow us to come up with new JAK inhibitors to treat different diseases, particularly for, and capitalize on that for topical drug uses. From the chemistry perspective, we understand the unique 3D structure of TOFA contributes to its overall potency, that out-of-plane aspect contributes to its potency, as well and more importantly, its selectivity when you look at things. And it's important to be able to get to that kind of kinase shape. We also, I've also shown that you can take advantage of one residue that's different in JAK3 versus the other JAKs and actually come up with a compound that can be truly selective for that compound. And so finally, in closing, structure-based drug design can hopefully influence the discovery of JAKs as well as other kinases as we go forward. So thank you.